Dear students, Namaskar. I'm Dr. Nutan Prakash Vishwakarma from Department of Biotechnology, Atma University. As we have discussed the concepts of sequence alignment, if you are new to this particular video, you just go back in the playlist and try to find the different parts of sequence alignment. Sequence alignment part 1, part 2, and part 3. In those videos, I have already discussed about what sequence alignment is all about, what are the applications of it, and what are the different algorithms we use to solve the problems of the sequence alignment. So in the sequence alignment algorithms, we have discussed about the dot matrix analysis. The limitation of the dot matrix analysis was we were just giving the visual representation whether the two sequences are similar or not. Means it's a kind of the qualitative analysis only you can make. So to overcome from this, different algorithm schemes that was known as dynamic algorithm, and for the dynamic under the dynamic programming there is two algorithms I have discussed. One was the Needleman-Wunsch algorithm and the second one was the Smith-Waterman algorithm. So the important limitation of the dynamic programming was it was so slow. Just to make you understand, I am giving you an example. Suppose today in your laboratory, you have isolated a protein, you have gone through the sequencing. Now you have the sequence with you. Now if you want to know that my protein, which one I have isolated in my lab, is showing the similarity with which sequence which is already available in the database. So suppose, for example, in database currently 6 lakh protein sequences there. So if you are using the Needleman-Wunsch algorithm or Smith-Waterman algorithm, either you go for global alignment or you go for the local alignment. How many times you have to repeat the things? You have to make the alignment 6 lakhs time. Do you think it is practically possible to conduct to go for the alignment 6 lakh time, and how much time it is going to take? The ultimate aim of the bioinformatics is to speed up the research in the right direction with a minimum span of the time. So here, if you are repeating the things, if you are doing the sequence alignment 6 lakhs time, definitely it is going to bog down the speed of your research. So now what is the solution? How we can quickly align the sequences with the minimum time? Now we have the solution that is known as word or k-tuple method, right? So whatever work you have to do 6 lakh time, now that result you are going to get with the help of the word or k-tuple method, you are going to get result within 1 or 2 minutes, depends on the internet speed and the load on the server, but not more than that. So let's see what word or k-tuple method and what is the magic behind it. Why you have to do the 6 lakh times with the Needleman-Wunsch algorithm, and here you have to make a single click and you will get your result in 1 or 2 minutes, right? And situation is going to be really worst once you have the gene sequence with you or DNA sequence with you, right? And if you go in the nucleotide databases, then the millions of sequences, billions of sequences are there, and the length of the gene sequence is too much. So definitely it is going to be near about impossible to find the homologous sequence. So how word or k-tuple method is rescuing us, is helping us to do the sequence alignment in a very quick way. Let's understand by word or k-tuple method. So previously before the blast, the computer science was there, principle was there, but so much no, applications of computer science used first time while scientists were developed the database, the program that is known as BLAST. So BLAST is the example of word or k-tuple method. Word method is for BLAST and k-tuple method is for the FASTA. Regarding FASTA, I will make a separate video and then we will discuss what FASTA is all about and what are the merits and demerits and shortcomings are there in the FASTA. So what are K-tuple method? The BLAST is, the full form of the BLAST is basic local alignment search tool. How to use the BLAST, I have made a separate video. If you go in the playlist, you will come to know how to interpret the result, how to perform the BLAST analysis and all. Practical, I have made a separate video. Here I am going to discuss the theory behind it, the algorithm behind it. So what are K-tuple method? Word on the word method, the BLAST is based and K-tuple method FASTA is based. Alright. And both are combinedly known as heuristic algorithm. We'll let you know what heuristic algorithm is all about. So before BLAST alignment programming, use dynamic programming algorithms such as the Needleman-Wunsch and Smith-Waterman algorithm that required long processing time and the use of supercomputer or parallel computer processes. And BLAST was developed by Alstul et al. In 1990 and has become since the standard in the search and alignment tool. So BLAST is basically being hosted in the NCBI, National Center for Biotechnology Information. Now the important question is, very important question is, does internet is required to use the BLAST in all the times? Answer is no. If you are using with the online databases, means you have the query sequence and you want to know that whether your query sequence is matching, showing the similarity with the sequences being submitted in the database in the public domain, you can go, definitely you need the internet. But what? But suppose you have your own proprietary database in your computer, you have developed own database and you are not making it publicly online. Or any company have developed their own database and they are not making it public. They are using their for their own research. Still, they can use the BLAST. BLAST is available over the web. You can download it and you can configure it with your own database, the personal database. And BLAST is going to give you result as you are getting the result in the online BLAST. So BLAST can be used online as well as offline. All right. So, what BLAST actually does? BLAST is a program that can detect sequence similarity between a query sequence and sequences within a database. The ability to detect sequence homology allows us to identify putative genes in a novel sequence, right? If you have any novel sequence with you and you want to identify whether it is a gene or not, you can use the support of the BLAST. It also allows us to determine if a gene or protein is related to other genes or proteins. And BLAST is popular because it can quickly identify regions of local similarity between two sequences. More importantly, BLAST uses a robust statistical framework that can determine if the alignment between two sequences is statistically significant or not. Right? So, the heuristic algorithm, I told you what about heuristic algorithm. Why word or K-tuple method is known as heuristic algorithm. Right? Heuristic algorithm is an algorithm that provides almost the correct answer, or a solution for some instance of the problem. So heuristic algorithm is an algorithm which gives you almost correct answer, not 100% accurate, but Needleman-Wunsch algorithm gives you 100% accuracy. Then what is the reason? Then if the heuristic algorithm means word or K-tuple method means BLAST is not giving you 100% accurate result, then why should we use it? So here is the comparison we can make. Suppose, let me give you one example. Very common example I am giving you. Suppose you are very hungry, right? You are very hungry. You came from the university or college and you are hungry and you demanded your mom to cook, to give something to eat. Mom have given you two answers. Number one, two options. Number one, I will give you the best recipe in the world, but you have to wait for one day. And second option is the remaining breakfast. What morning your mom cooked, that is left, kept in the fridge, you can have it right now. But that is not so tasty. And you are so angry. You are so hungry. What you will do? You will not wait for a day so that you can have the best recipe. What do you want? You will have the remaining breakfast and you will satisfy your hunger. The same thing happened in the case of the research also. Suppose you have one sequence and you want to know with which sequence in the database your sequence is showing the similarity. If you go for the Needle-Manus algorithm, you have to make it for 6 lakh time, or if your gene sequence, you have to do the experiment 1 crore time. Maybe 100 days you will get to get the result, the best result. But if you use the heuristic algorithm, you will get almost the accurate result with few mistake possibilities there, and that will be cope up with the statistical method. That's why whatever is not possible in 100 days, that you are going to get quickly in 2 minutes, right? That is the miracle of the heuristic algorithm. So a lot of versions of the BLAST is available. The important version of the BLAST is BLAST P. Here is the very important things you have to remember. If you have the amino acid as a query and you want to match it with the amino acids like protein as a query and proteins in the database, you can use the BLAST P. If you have the nucleotide as a query, means the DNA sequence as a query, and you want to match it with the DNA sequences available in the database, you can use the BLASTM. So here you don't have to mug up anything, very simple. As name suggests, BLASTM is protein and protein as a query and protein in the database. BLASTM query is a protein nucleotide and nucleotide in the database. So here you don't have to remember much in the BLASTP and BLASTM. Regarding the BLASTX, if you have the nucleotide sequence and you want to know that what may be the putative protein, what may be the possible protein it is coding. So once you call that particular nucleotide in the BLASTX, and if it is coding, BLASTX automatically will translate it in the 6 reading frames and it will match with the protein database. So if you have the nucleotide as a query and you want to match it with the protein sequence database, you can use the BLASTX. And if vice versa is there, if you have the protein, you want to match it with the nucleotide, you can use the tBLASTM. So that you can remember. These are very basics variants of the BLAST. Although a lot of variants are there, I am going to discuss few of them. So in addition, NCBI has some of its own specialized variants of BLAST, for example, MegaBLAST. So MegaBLAST is a program that can rapidly complete searches for sequences with only minor variations and can more efficiently manage queries with longer sequences. If you have the longer sequence, MegaBLAST is for you. MegaBLAST will increase its word method. I am going to tell you what word method is all about. PSI and PHI blast are more other powerful blasts too that allow more complex and evolutionary divergent protein to be aligned. Right? If your protein is too much diverged and your normal blast is not giving you result, in that case, if you use the PSI and PHI blast, it have more good algorithm to detect the complex and evolutionary divergent proteins. So, through a heuristic approach, BLAST identify homologous sequence by locating short matches between the two sequences being compared. And this process is referred to as seeding, and it is after the initial match that BLAST begins to make local alignment. During the process of seeding BLAST, I'm going to tell you about in the algorithm what seeding is all about, right? So let's understand by the algorithm. You can pause the video and you can refer the notes which I'm going to provide you, and you can understand directly from here only. So how BLAST works so efficiently and so fast? Let's try to understand. Suppose you have a query, the protein query. You want to match it with the database with the sequences of database. So what you can do in this case, once you have the query as a protein sequence and you put in the BLAST break it in the 3 uh, letters, just like here see the example. In this context, a word is simply defined as a number of letters. For example, BLAST P, the default word size is 3, right? Suppose you have a query A, B, C, D, E. If you have a query A, B, C, D, E, suppose this is simple. This is the protein sequence. This is the A, B, C, D, E. Once you will put in the BLAST P, what BLAST is going to do? BLAST will make the 3, just like if you have read the ORF, open reading frame, in that case we can make the codons, right? So likewise make the codon type, like ABC, like it makes the ABC, BCD, CDE, like that. All right. Then, and this is known as seeding. Word size, suppose if word size is 3, So while you are using the BLAST P, the word size is actually 3 only. And if you have the nucleotide sequences, the word size is 11. So whatever query sequence you have, it is going to make the in the 1111 group. And that 1111 group, suppose here I am giving the example for sake of simplicity, I am giving the example of the protein. So if you have the protein sequence, suppose this is the database with you, right? This is the database. And whatever query you have put it, that query individually three amino acid sequences program is trying to identify where is actually does your sequence in the database is showing similarity with these three codons, these three amino acid bases, and then it will make the list. It makes the list wherever it matches. Just like no, you have the PDF file and you are doing the control F find, right? If you have the word, if you have the book like a Leninger book and you want to read about the amino acid, so simply put the control F and put amino acid. So out of lakhs of crores of words, it is going to make you highlight the amino acid. All right, the same way it is just doing the same kind of the work, the find tag, control F kind of the work. You can just take the analogy to understand. So for each word from the query sequence, find the list of words that will score at least there is the some threshold. If not exact match, if similar kind of the amino acid is matching, it is going to report you. All right. Because in the where you are making the control F no in the word file or in the PDF, it is going to give you the exact result. If exact match is there, then it will highlight. Otherwise, it will simply say that it did not found. But in the word, if it is not exact match, if it's similar amino acids there, still it is going to report it. So that is known as simply seeding. At least you found some hits in the sequence. So for the query, find the list of high scoring words of length W. Then compare the word list to the database and identify exact matches. So these are the different, whatever the list you have made of the 1111 bases in the case of the nucleotide, and if you are taking the protein as a query, the three matches, three amino acid matches. So compare the word list to the database and identify exact matches, and then what it do, wherever you get the match, wherever you get the match, then it start matching, it extending alignment in right direction and left direction, both the direction. And until it giving the alignment as a positive, means if the alignment score is increasing, it keeps on extending, right? I'm telling you the example. I'm giving you the example. Suppose if you are traveling with the train in the suppose in the general compartment, what happened? Whenever whenever train no depart, people are standing in the queue, and then they request the fellow passengers, those who got the seat, that please give me some space to sit down. Previously you request, then passengers sit down, and slowly as train pass, slowly as train move, now they start expanding. Isn't it happen? The same way, once you got the hit somewhere in the database, in the sequence of the database, once you got the hit, now you can keep aligning in both the direction left and right, and until the score is increasing, you blast keep uh, aligning the sequences, and whenever the score goes starting down, because whenever the mismatch, definitely you know the score is going to be in minus. So from this particular threshold, I'm teaching you in a very nutshell. This video is I'm making for the MSc Biotechnology or MSc Microbiology or BSc Biotechnology students, those who are not much aware about the algorithms and the computer science, Parnin, etc. So I'm teaching you the blast algorithm in very nutshell, very in the crude. I'm not using much terminology, the technical terminologies. So until your score is increasing, it keeps aligning, and whenever it starts decreasing, blasts stop the alignment, and then it makes the ranking and top 100 most aligning fragments. It shows the result. So that's how you get the result very quickly. So suppose you have 6 lakh proteins in the database, it is going to make you only top 100 or top 20 or top 50 as you set in the settings. You will get there. Okay. So how to use the blast? I told you this is the very simple interface of the NCBI, right? So in the NCBI, here you can find the blast, the link of the blast. Then if you click on that particular link, you will get the different versions of the blast like nucleotide blast, blast XT blast, and protein blast. Other variants also like smart blast. It find protein which is highly similar to your query. Primer blast. Suppose you have the any particular sequence you want to design a primer, PCR primer. You can use the primer blast. In the global align, we use the algorithms of the blast, and you can go for the global alignment as well. CD search, you can search for the conserved domains in your sequence. GEO, through GEO you also can use the gene expression profile. IG blast, if your work is related with the immunology, you can use the IG blast. The VEC screen, if you want to search any contamination present in your sequence after sequencing. So CDART, regarding the structure, 3D structure of the any domain, you can find through the CDART. So there are a lot of specialized searches like target loci. If you are searching for the markers for phylogenetic analysis, multiple alignment you can do after align sequences using domain and protein constants. Bioassay, you can search protein or nucleotides targeting PubChem bioassay. Mole blast, you can establish the taxonomy for the uncultured or environmental samples, means metagenomics. So these are different specialized searches available. So how to perform the blast? First you have to identify the query sequence, obviously whether you have the gene sequence or protein sequence. Then you have to use the algorithm tool, means whether you are using the fast or blast. So suppose in this case I am using the blast. Then select the database from which database actually you want to match your sequence, right? So protein or nucleotide sequence databases, you want to match your sequence with all the databases which is publicly available or with a certain database that you have to choose. Then you hit the button fire as a query, and whatever result comes, that you can analyze that whether it is statistically significant or not, or it is chance finding. So how to perform the blast? I have made a separate video. How to interpret the result of it? You can go in the practical session. There is the explanation of the e-value and all. There you will come to know that whether your result is significant or not. So this was the very simple theory behind the blast. So next video I'll make on the FASTA, and I will compare also among the blast and FASTA. So if you like this video, if you understand something, please hit the like button. You can subscribe and share the buttons also. And so that's all for today. We'll see you in next video very soon. Till then, take care. Thank you very much. Namaskar. Jai Hind.